There have been quite a few advances, I suppose, in using embryonic stem cells in a way that we might not have thought we could do beforehand. For example, these 3D cultures that we're doing. Even though people have done 3D cultures before, it seems like now we're doing very well to push that a little bit further.
The progression of 3D biology was driven in a lot of ways by induced pluripotent stem cell technology becoming available and widely used. Being able to do disease modeling in stem cells and then create a 3D model has really, I think, driven a lot of the 3D market adoption.
Until now, we've really only been able to use animal model systems as a proxy or as a surrogate for human development, but being able to use a pluripotent stem cell is a very intriguing area to have new insights to actually studying human-specific development.
I mean, can you imagine being able to take a patient-derived disease-specific cell line and be able to grow them up, and that this model has the same response as the human? I mean, that's precision medicine. Can 3D model be used for precision medicine?
The fact we're now at a way where we can use this system to look at early aspects of development, I think is quite fascinating. But also the organoid field in general is quite fascinating, because now we have sort of opportunities to look at how organs can develop as well.
Prior to some of these 3D cell culture models, studying these adult stem cells in a dish was virtually impossible. And so there's lots of labs who are exploiting these organoid models to study adult tissue development in a way that we've not been able to do before.